From the Vulcan mind meld to Professor Dumbledore's Pensieve, there are countless ways in which people share memories in fictional films and TV shows. But such fantastical ideas could soon become a reality, using electrodes implanted in the brain. Neuroscientists have already begun trialing implants that boost memory loss, and in the future they believe these implants could be used to replicate memories in the brains of others. Research teams from the University of Southern California and University of Pennsylvania have been testing the technology on epilepsy patients. These patients already have electrodes implanted in their brains, which means the experts didn't need to insert the prostheses in new patients through risky brain surgery. The research centered on the hippocampus, a seahorse-shaped part of the brain associated with the formation of memories. The hippocampus gathers sensory information that is then transformed into short-term memories, between 15 and 30 seconds. These can then form more lasting memories, but only if they are accessed while the hippocampus is storing them. People with significant memory deficits typically have a damaged hippocampus. The USC team, led by brain implants expert Ted Berger, was interested in two particular areas of the hippocampus, called CA3 and CA1. In particular, the team thought that an electrical signal traveling from CA3 to CA1 was key to memory formation. Therefore, they tried to recreate a similar signal in order to restore the hippocampus functionality. To do this, the researchers monitored the brain of 12 epilepsy patients performing a memory exercise that included memorizing pictures to see how CA3 and CA1 interacted. Eventually, they developed a mathematical model to predict the pattern of the signal CA3 would fire to CA1. The predictions were correct 80% of the time. The USC team's idea is that brain implants could provide electrical stimulation resembling that key CA3 signal to improve memory in patients with hippocampus damage. The work builds on a previous study carried out by Dr. Berger in which his team taught rats to learn a task, pressing one lever rather than another to receive a reward. Using embedded electrical probes, they recorded changes in the rat's brain activity between the two major internal divisions of the hippocampus. During the learning process, the hippocampus converts short-term memory into long-term memory. Dr. Berger said, "No hippocampus, no long-term memory, but still short-term memory." Researchers blocked the normal neural interactions between the two areas using pharmacological agents, leaving the rats no longer displaying the long-term learned behavior. The rats still showed that they knew when you press left first, then press right next time, and vice versa, Dr. Berger said. And they still knew in general to press levers for water, but they could only remember whether they had pressed left or right for 5 to 10 seconds. Using a prosthetics model, the researchers then developed an artificial hippocampal system that could duplicate the pattern of interaction between the two internal divisions of the hippocampus. Long-term memory capability returned to the pharmacologically blocked rats when the team activated the electronic device program to duplicate the memory encoding function. In addition, the researchers went on to show that if a prosthetic device and its associated electrodes were implanted in animals with a normal, functioning hippocampus, the device could actually strengthen the memory being generated internally in the brain and enhance the memory capability of normal rats. What is the hippocampus? The hippocampus is an area at the back of the brain, housed in the medial temporal lobe. Its name literally means seahorse, as it has an elongated and slightly curled shape. It is in charge of transforming sensory information into short-term memories, which it stores for about 15 to 30 seconds. Only memories that are accessed during this storage phase are later transformed into lasting memories. According to the USC study, the electrical interaction between two sections of the hippocampus called CA1 and CA3 play a decisive role in long-term memory creation.